I'm working at Weill Cornell Medicine, Qatar. So it's a branch campus of the New York University Weill Cornell. Here's our building. And the reason why Cornell is in Qatar, it's actually because Qatar has a very dire health problem. The diabetes numbers in Qatar are actually the largest in the world, with 23% of Qatari adults having developed diabetes. And within Cornell, we're actually researching what is the reason for complex disorders, in particular, of course, diabetes, the comorbidities, and ask what makes us different. So the different reasons why we are all different. I mean, we know very well it's our genes, like what I showed on here, but there's also the diet, what we eat, whether we smoke, our lifestyle, whether we are physically active, the drugs we take, and the gut microbiome. All these factors, they influence different health outcomes. So I just show here a few of them. So especially the cardiovascular disease, diabetes, Alzheimer's, cancers. I mean, they're all very much intertwined. And they are linked all together through what actually happens in our bodies. So what I show here is the paradigm of biology. You go from DNA to RNA to protein. And over the last years, this has been actually mirrored by a boost in technology. So you have genomics, you have transcriptomics, you have proteomics mirroring all these different steps. And then, I mean, I think that's why we're here today. It has been continued by a part of what these proteins do, transporters, enzymes, they produce metabolites. And then I think we shouldn't forget there's also other omics, the regulatory part, epigenomics, microRNAs, protein modification, glycomics, and you have it. Here in my talk, I will talk especially about metabolomics. And metabolomics is particularly interesting because I think it's really the wiring and what makes our body tick. So I show here a pathway map. And just very quickly, I think in this community, it's maybe you have seen it already before. I mean, it's really like we try to measure as many metabolites as possible and then connect them to the disease in endpoints. And I always like to mirror this like more from an engineering point of view to say basically what happens in our body is a complex piping system. And I will come in a moment, especially why I'm designing these valves in here. So yeah, there's basically a flow through the system of the different metabolites. Now, this is a cohort event, and I think the very important concept to never forget, why would we go for very big cohort studies? And that's actually because we can experiment on mice, but not on humans, but nature can. So in the end, human population cohorts are probing existing variation that's there, created by nature. So in a way, it's experiment conducted by nature without IRB, by the way. And what we leverage is links between these different factors. So you have the genome, microbiome, and all the factors that in population studies, we try to link to the risk factors. So you may know of Framingham, where years back, it has been shown that smoking, for instance, is giving you a higher risk of a heart attack and so on and so forth. And for instance, in Germany, also have the CORA study, which I have been working a lot with as well. Now, the problem with these studies is, of course, they can identify a lot of risk factors through association, but it's very hard to actually get detailed functional understanding out of that. And I think a key idea at some point when the omics technologies really took off was to say, we put what we call deep molecular phenotypes as an intermediate phenotype in the middle, like the ones I showed, epigenome, transcriptome, until the microbiome. So you measure as many parameters as you can in these populations, in blood, urine, saliva, whatever you can get your hand on. And then you don't connect, I mean, you connect, of course, directly also the phenotype to the endpoint, but in addition, you can now connect all the different phenotypes you have, plus the genotypes, through these intermediate traits, and then from these intermediate traits, do association to the endpoints. And that has been shown to be very efficient because the strengths of association between these different parts are much stronger and also much more concrete. So there's of course hundreds and thousands of intermediate traits and not all associated with everything. So you get a much finer picture. I think biocrates came up with this image very early on, like saying it's like an imaging, like the more traits you put in, the better the image of your participant or your patient. And then of course the power, once you start putting omics platforms into these cohorts, the more you put to them, it actually explodes exponentially. You can say, I can do one epigenome against the transcriptome, against the proteome, and so on and so forth. So whenever I get another layer, I would actually get multiple layers of comparison and study. So in the end, this technique reveals really new pathways and can indicate new therapies. And in the remainder of my talk, I will show you a few of the examples how actually we use that in the past to use genetic variation in the human population to figure out how that influences changes in our metabolism. All right, and I think this is a paper, biocrates will like this, but it's not for me, it's not an advertisement, it's the truth. The first GWAS we ever conducted in 2008 with my colleagues in Munich, especially Christian Giger. And you might have seen earlier the talk of Jurek Adamski, he was also on this paper. So the first ever GWAS with metabolomics has been done with a pretty small study, but has been done with the biocrates platform on only 280 samples. So we see later now the dimensions where we are today, but this is the first time I got into contact with metabolomics and genetics combined. It was really an eye-opener. And I want to try to share that a bit with you. So I think now I'm taking things already before, and we didn't know that when we started, but the concept of what we now call the genetically influenced metabotype, some people say metabolic QTLs, but I think the genetically influenced metabotype is something bigger. It's more the combination of multiple metabolic QTLs. So the idea is to say, in your population, you have, for any given variant, and there are millions of them, you have for every person a state. They could be major allele homozygote, basically white type for a variant. They could be heterozygote or minor allele homozygote. And then we associate these variants against all metabolites and try to figure out, are there any associations with a metabotype or with a metabolite? So what we found, and here's the prototype from my point of view for metabolically genetically influenced metabotype, it's the association of a variant in an enzyme, the FADS1 enzyme in this case, which has the function to desaturate the C23 to the C24 arachidonic acid, fatty acid. And what we found in that study is there was a very strong association basically with this genetic variant and the throughput of that reaction. So in this case, it was so strong that basically major allele homozygotes were 50 times faster in producing arachidonic acid than minor allele homozygotes. And then, of course, you can imagine what impact that has. And also that these are really very, very frequent genotypes. So these are not monogenic disorders. So we tend to see these nowadays as, in a way, like valve settings. I showed you before this kind of a flow chart, the piping, and what it means is that your internal metabolic flow is not the same from person to person. It's different, and depending on this setting, everyone is individual in their capacity of metabolizing different inputs, generating different outputs, clearing toxins, and so on. And what we want to understand is how this combination of these different valves actually influences our health. So here I show again, just to make you aware of that. So we're talking about frequent variants, not rare variants. So for instance, for FADS1, you would expect in 100 people, and I think that's about the listenership we have here. Oh, we have much more at the moment. So I think within the listeners, there would be at least 5 minor allele homozygotes. And I think they are all pretty healthy, but they may have a very different phenotype in what they can do with their arachidonic acid. And in combination with other variants, that could have very interesting results on health risk and so on. So what we just call them is like, I mean, you know that from pharmacogenomics, fast metabolizers, medium and slow. I mean, I'm a physicist, and I still have to show this slide, although I already did very many times. So actually, I didn't know Archibald Garrett. So this is Archibald Garrett, and he's the inventor of inborn errors. Biologists know him very well. So he rediscovered the Mendelian laws of inheritance for metabolism. And he was saying that basically inborn errors of metabolism are merely extreme examples of variation of chemical behavior, which is probably everywhere present in minor degrees. So that's actually what these frequent variants that change metabolism are. And he already 100 years ago basically suggested that these variants, this what he called chemical, or nowadays I would say metabolic individuality, is actually predisposing us to complex disorder. So he had already, although he invented the inborn errors of metabolism, so the strong diseases, he already invented also the idea of complex disorders and the link to many variants. All right, so having understood the role of this, we then set out to basically map out what's out there, the Atlas of Human Metabolic Individuality, by identifying, well, all relevant genetically influenced metabotypes. And I think why we're here today is, of course, we want to do something with this in these cohort studies. So we want to know how can we translate this knowledge once we generate it to clinical application and biomedical application. So here is where we started. This is the lab of the Helmholtz Center, the one that Jurek Adamski set up, and with the Helmholtz Center and cooperating with different metabolomics companies, we actually set out to identify these different genetic variants. And here is the initial timeline from the initial studies we did. So the first study here is just very low powered, the ones I just mentioned, the first one with the biocrates. And then we went on, we had this bigger study where we actually used the first kids from biocrates. Then we went on, looked into urine with NMR and the genomics company. We worked also with non-target metabolomics with Metabolon. And I think at this stage, the numbers really already crept up. So we're at 2,800 samples, 250 non-targeted metabolites. We're pretty lucky at that point to get that paper into Nature. And what we identified were 37 of these different metabolically determined or influenced metabotypes. And each dot here is actually one of them. FATS1 is over here, and each one of them actually tells their own story. And I have a talk where I really get into all of them, but that would take hours. So I invite you to read the paper if you want. Just a quick wrap up of this. I think one key finding was that many of these genetic variants actually really linked directly to the genes that were enzymes or transporters of the associated metabolites. So there's a direct link between the gene and the function. And this can be used in multiple ways. If you don't know the function of the gene, you can make a hypothesis on the gene. If you don't know the identity of the metabolite, you can make a hypothesis on that. And you can then overlay these genetic variants with disease. So remember, this was 2014. So GWAS were still just going on. So overlaying GWAS, disease GWAS to metabolomics GWAS was also in a way a first. And what we really found is there was a very large overlap. And I think this map must be much denser now between these disease loci, risk loci, and also other genetic variants and metabolic variants. And that again repeats a bit this idea of saying they're intermediate phenotypes. So you have, for instance, a disease GWAS on cardiovascular risk, and that overlaps with certain lipids. So you can understand, and I think I'm going to have that as my next slide. I can actually understand the different pathways that are involved in these diseases. So we went on, we worked with other studies on unknowns, we worked with NMR. So this is a little bit like the technology the company Nightingale is doing as well. And then we really went to, well, I think the biggest mGWAS so far until very shortly, where we did a study on almost 8,000 individuals and determined an atlas of genetic influence of human metabolites. And that was published in Nature Genetics 2014. And so this is basically the same figure as before with these tiles, but this time we have 145 of them, so I cannot put them onto tiles anymore. What's interesting here, again, you can classify them in what overlays with inborn errors of metabolism. So many of these variants are also moderate forms of more severe diseases. There's an overlay with many drug-related loci, so very much interesting for drug development. I will repeat on that later as well. And then very many overlaps with disease as well. And then in terms of classification, there are enzymes, transporters, and really only very few where we didn't know the function. And that in the meantime, some of them even have been characterized as enzymes like PROXD2. And what you see here is now what we call the atlas. So it's actually a network of partial correlations and links to genetic variants. So you can use this network now to navigate the atlas. And I think I mentioned I wanted to show you an example of a pathway. So this would be actually a zoom into this network. So the dots would be different metabolites, and the colors are GWAS associations to blood pressure, cholesterol levels, blood coagulation. And as a whole, you see here different genes that play different roles in blood pressure regulation or cholesterol level and so on. And that means having all these data from all the individuals in this population studies, having them overlaid with a lot of metabolomics data, we can create and reconstruct actually knowledge that can put you further in understanding how these pathways works and where eventually could intervene. Like for instance, here you see the ACE enzyme, which is actually a target of blood pressure lowering drugs. All right, and then I said until recently it was the biggest one. So this paper is now in press. I think it should be come up very, very, very shortly. It's a study led by Adam Butterworth and Claudia Langenberg, and the hard work has been done by Praveen Surendran and Isabel Stewart. And I think very strongly in the background was Gabi Kastenmüller's work, and I think she's talking later about really integrating all this data into really very user-friendly web interfaces. And what you would then see, I think the paper will be coming out in the next few weeks, that now we don't have just one variant goes with one single metabolite, but you can now see that the strengths of having this metabolomics and not just one metabolite. And what you see here is a bunch of metabolites, a metabotype, if you want, that are associated with multiple variants at the same locus. And you see, as in genetics, you have these kind of correlated or even uncorrelated variants that you can then find met. You have rare variants and frequent variants that all influence these metabolites, but not in the same way. Like for instance, you see one variant that influences everything, some positive, some negative. So you can really make hypotheses about what are the products, what are the educts, what is regulatory, is it a variant that changes the enzyme function or more its expression, and so on and so forth. And this kind of stuff could then really be used to identify individual drug targets, interpret them, and get to potential modes of action. Okay, and now, so this is now for the last part. So this is something I did very recently. So the UK Biobank now has 500,000 individuals, and I think they go the same way as the other smaller cohort studies. I mean, most of what I showed you was Cora, twin study, other studies in the UK. Well, UK, here, this is, I think, in my view, really the biggest and most open and best biobank that we have on the planet, UK Biobank. They have 500,000 individuals, and there is a project to do NMR metabolomics at the moment on them using the Nightingale NMR platform. So they are measuring around 200, 250 metabolic markers, many of them lipoprotein properties, a few metabolites, a few proteins, measured in blood. And at the moment, there's data for 120,000 of them. The idea is that there would be another 300,000, not another, there would be a total of 300,000 samples available in 2023, next year. And when the project is finished, the whole cohort of 500,000 shall be covered. And now imagine that you would have this kind of data combined with, at parallel, they are doing analysis on the Olink platform for proteomics. And imagine you would have other more detailed targeted, non-targeted metabolomics platform on them, methylation data, whatever. You can get transcriptomics data that we're working on. So imagine what you can get when you put all that together. I recently presented use of that data on the DNA Nexus platform, how you can get at the metabolomics data through the platform. So there's a very democratic way actually of sharing all this data. It's very open. We published that on bioarchive. So here's the link. And I think this is really the last slide I really want to insist on. On the x-axis, you have all different variants from one study where they identified the lipid risk loci in a very big study with 1.6 million individuals. And from these, I picked 141 that really replicate fully on this NMR platform. And you see to the left the p-values and to the right the beta values of the association, the effect size. So each vertical line is actually an association profile with all metabolites. And imagine how this would explode if you would not have just the NMR traits. You see they are highly correlated here, but if you would have even more detailed metabolomics data here. And down to the right, you see examples of these association profiles. And to the left is actually an example of how this could really be used for drug development. So this is the variant in HNGCR, which is the starting target. And what you see here is actually proves you on metabolites that genetic inhibition of mHGCR is actually really modeling what a starting drug would do. So now I could take any other drug target in here and then query, say, what would the impact on metabolism will be, the entire profile, and what would I expect from that inhibition? I can plot that against the outcome, myocardial infarction. So here, for instance, you would see the effect size of statin against whether or not individuals had an incident myocardial infarction. And overlain in size is the Mendelian randomization p-value to show you what are the causal ones. So what you can see here up there is actually the action of statins on LDL, which also is shown to be causal for MI. And then you can do the same thing and ask more. This just as an example, nitty-gritty questions. Being a vegetarian, what's the impact of being a vegetarian on the metabolic profiles, or eating processed foods? And here you see basically the effect of being a vegetarian on the lipids that actually give you a risk for an MI. So being a vegetarian apparently is not a bad thing for your health. All right, so that brings me through the thing. Through my talk. So just to wrap things up. So why are we so interested in having so many data on so many samples? And I think I showed you all these different things you can learn, the match between metabotype and gene function. You can do a lot of functional genomics, functional metabolomics there. The effect sizes are so large that you can do basically see how combined variants are affecting your health. They are frequent genotypes, so it's not about rare variant, it's about basically precision medicine. The overlap with the GWAS loci interpretation of disease GWAS. They are intermediate phenotypes to disease. And then, as I said, if with the power increasing, you can do gene-gene interaction, gene-environment interaction. So the more you have, the more your statistical powers. I cannot repeat that enough. You cannot do a study. I mean, we did the first in 280 people, but the higher your statistical power, the more you get out of that. And nature has hidden a lot of weak associations there, which are weak in terms of changing the metabolite, but not weak in terms of information content you can pull out of that. So even a very weak functional association with certain metabolites can give you a very big breakthrough in understanding functional genomics and drug target validation. So most recently, I think I just name drop them here. Of course, you can now use metabolites as intermediate traits for Mendelian randomization to actually show they really or determine whether they're really causal, which also would mean like targeting certain metabolites through some therapy, whether it's meaningful or not. Not really metabolites, but an example would be targeting LDL is a good idea. Targeting HDL, probably not. I mean, that's the founding example of Mendelian randomization. And then polygenic risk scores is also really coming up. You can do polygenic risk scores on metabolites as well. And with that, I'm through. So if you're interested in GWAS with metabolomics, I have a blog where at the moment there are almost 80 metabolomics GWAS registered. So you can really get all these directly to these papers. And I would like to thank my founders, especially Qatar Foundation, my team of course, my team members, and especially the people involved in all the GWAS over the years, my team members here in Qatar and elsewhere. And with that, I thank you and hand back to Matthias.